Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. In the last class, we started talking about multiple hypothesis testing correction. We talked about why this type of correction is required. We have seen that when you are testing a large number of hypotheses, the number of false positives can increase, and we have started discussion on correction methods, right. So, how we can correct or control for these false positives or the type 1 error. Okay. So, in the last class we mentioned about family wise error rate correction method, and in this class we will be talking about FDR correction method. So, once we have discussed this FDR correction method, we will move on to something called the interpretation of the results that we obtain from DGE analysis. Okay. So, this is the agenda for today's class. So, first we will discuss about FDR correction method, and then we will talk about interpretation of results from DGE analysis, right. So, we generated this table from DGE analysis and how we actually proceed with that result. Okay. So, let us start with the FDR correction today. So, the first keyword that we will see is the adjusted p value, okay, and you have come across this term before, and the second keyword we will see is gene ontology. Okay. So, briefly we have two types of methods for controlling type 1 error when we are doing multiple hypothesis testing. So, the first type is the family wise error rate correction, which we discussed in the last class, and then the second type of method is the false discovery rate correction or FDR, which we will be discussing in this class. Okay. So, let us start with this FDR correction method, and what you have seen in FWER correction methods is that we are worried about making even a single type 1 error, right. So, that is why we want to control alpha at the same level even after 10,000 tests. Okay. But in many real life scenarios, what you will see is that we are not really worried about making even a single type 1 error, and you can sometimes accept a certain number of false positives. Okay. It is not that we want a lot of false positives, but we can tolerate certain number of false positives in real life situations, right. So, in our example, let us say when you are doing this differential expression analysis, we identify 100 genes as differentially expressed, and imagine if let us say 3-4 genes are false positives, right. So, if you remember this table, truth table that we talked about, right, we have the actual truth and the statistical decision, right. So, if only 3 or 4 genes are false positives, then that is not going to bias our analysis that much. Okay. So, in that case we can tolerate, right, a small number of false positives, they will not substantially change our results. Okay. So, that is what I state here, right. So, we can probably control the type 1 error at a certain value, right, rather than trying to control it at very stringent cutoff, right. So, we can perhaps tolerate maybe 5 to 10% false positives, again depending on the test or depending on the inference that you are making, we can tolerate this 5 to 10% of false positives in our data. Okay. So, this is actually the approach that these false discovery rate correction methods take. Okay. And there are different methods that are out there. The first one and the most popular one is the Benjamini-Hochberg method. You also have other methods, for example, Benjamini-Cottelli method, and then you have Story-Tipsiani method. This is a Q value method, it is called the Q value method. So, we will talk about the first method, Benjamini-Hochberg method. This is the most popular one, and the rest of them I will just give you the references, and of course, you can have a look. So, coming back to the statistical test where we talked about this type 1 and type 2 error, just to remind you, right, we have these situations, we have this actual situation of the truth, and then you have the statistical decision on the other side, and then we have also defined the false positives which are the type 1 error, then we have the false negatives, and of course, we have the true positives and the true negatives, right. So, the false discovery rate actually relates to these false positives and true positives, right, and is defined as the false positive number divided by the true positive plus false positive. Okay. Okay. So, let us now go into the Benjamini-Hochberg method, or sometimes you will see in many tools is written as BH method. Okay. And again we will take the same example that we took earlier. Okay. So, we have n hypothesis tests, right. We have this null hypothesis H naught 1, H naught 2, so on up to H naught n, and we have corresponding p values p 1, p 2 and so on up to p n. okay, and probability of type 1 error that in the original situation, right, in each test is alpha, right. Now, what we do in this method is that we order the p values first, right. Again, we have seen this kind of situation, right, in Hom Bonferroni method, where we actually order these p values according to their magnitude, right. So, the smallest p value will come first. This is the p 1, right, within bracket, right. Again, this is denoting these are sorted p values, right. This is not the original p values, right. So, the smallest p value comes first and the largest p value at the last, right, at the end. And we also have the ordered null hypothesis according to the sorting, right, which we call as H 0 comma 1. So, this is the first null hypothesis according to the p value sorting, right, and similarly we have up to H naught n. Okay. These are again the null hypotheses that are ordered against the p values. So, what we do in case of Benjamin-Hochberg method is we actually try to find the largest k. So, k is between 1 and n. So, n is the number of statistical tests that we are doing. So, that this p k, right, this is the ordered p value, is smaller than k divided by n times alpha. Okay. Now, this alpha we can set, of course, and we can say this to be 0.05 and so on, right. So, what we are doing is we are comparing these ordered p values now. Okay. So, if you remember that we have this p 1, p 2, right. So, since this p 1, right. So, this is the smallest p value. So, k is 1. So, what we do is we are comparing this with 1 divided by n times alpha. Okay. For p 2, we are doing this, comparing this with, right, 2 divided by n times alpha and so on. We keep on doing this, and for p n, right, this is the ordered one, we have n divided by n times alpha, which means we are simply comparing this with alpha. Okay. So, when we are doing these comparisons, right, and we do this comparison and we want to find the largest k for which we have this condition satisfied. Okay. So, p k is less than k by n times alpha, and what it means then for p k plus 1, these conditions are not satisfied, and so we can, what we can do now is then we can reject this null hypothesis H naught 1 up to H naught k, right, which satisfy this condition, okay, where they are less than the cutoff that is set for them. Okay. Again, the cutoff is adaptive, right, as you can see we are changing the cutoff based on the p values, right, and we do not reject this null hypothesis H naught k plus 1 to H naught n. Okay. So, what this method will do, right, it will control the error, right. So, we will control these false positives, right, number of false positives at a certain value. Okay. It is usually 5% or 10%, again depending on the alpha that we choose. Okay. And if you have seen this, right, once we apply this method, we calculate something called adjusted p values. Okay. And sometimes they are also called key values, but it could be misleading, again depending on the method that you are using, right. You should always look at which method has been applied for this FDR correction, right. This is something that is important. So, we either call them as adjusted p values or sometimes also q values. Okay. So, these adjusted p values are calculated from the original p values of the statistical test by taking this FDR rate, right, the false discovery rate into consideration. Okay. And I will give you the formula, right, how we actually calculate these adjusted p values. Okay. So, again going back, we have these ordered p values from n test, right. So, again from p 1 to p n, right, and what we do is we start from p n, right, this is the last p value or this is the largest p value, right, and we calculate this adjusted p, right, this is the adjusted p value. So, adjusted p I, this is the minimum of p I into n divided by I and or compare with p I plus 1. Okay. So, if I is the rank of that p value, right, we multiply that with n, that is the number of statistical tests divided by I, and then we calculate, also compare with p I plus 1, right, and take the minimum of these two. Okay. And so, n is the total number of tests and I is the rank of the p value, and in any case, right, if p I is greater than 1, we then set p I equals to 1, right. So, this might apply for the largest ones, right. So, p I, the adjusted p I might go above 0.1, right. So, these are actually adjusted p values. Okay. So, this is adjusted pi when you are calculating these values. Okay. So, this is how we calculate this adjusted pi, right, and what we now do after we have adjusted p values, if an adjusted p value is less than the FDR threshold, then we consider a gene to be differentially expressed. Okay. Now, what is this threshold? This is usually set at 0.05 or 0.1, right, and they correspond to 5% or 10% false positive rate. Okay. So, if we set this threshold to be 0.1, right, we can expect about 10% false positives in the list of differentially expressed genes, right. So, finally, we identified let us say 100 genes that to be differentially expressed, about 10% of them are false positives, okay, because we have set the threshold at 0.1. Now, if you want to make it more stringent, of course, you can change it, you can make it 0.05, 0.02 and so on. So, going back to this table, right, this is the results table that we looked at, right, and we have this column p adjusted, and this is actually adjusted according to this Benjamini-Hochberg method, okay, and these p adjusted values are calculated from these p values. What you will also notice is that this p adjusted is bigger than the p value, right, larger than the p value, right, and that is because of this adjustment. We remember we multiply with the number of tests and divide by the rank of that p value. Okay. So, you will notice this p adjusted is always larger than the p value that you get from the statistical test. Okay. One thing you should not confuse is that this list is actually ordered according to the gene names, right, it is not ordered or ranked according to the p value. So, once you have generated this list and this is done inside DSEC2, once you have generated this list, you have to sort the p values and then rank the p values, right, and calculate all these adjusted p values and put them back here. Okay. Okay. So, we have now talked about this FDR correction method, and you see, right, this is less conservative than the FWER methods, and they have more power in detection of the true positives. Okay. And this is why this is a method of choice now in case of differential expression analysis. Now, one question you might have is that which method should we choose, whether we choose FWER or FDR, right. Again, it depends, okay, depending on the question that you are answering. Okay. So, in situations where you can tolerate certain false positives and you are doing a huge number of tests, you probably want to apply FDR, and in situations where you have a smaller number of tests, maybe you are doing 10-20 tests and you do not want to commit or identify any false positive cases, in that case you will use this FWER methods. Okay. So, now we want to move into the results part, right. So, we have generated these results and we have understood all the components of the results, right. So, we have talked about log to fold change, we have got the p values, we have got the p adjusted values, and we now understand how these p values are generated, p adjusted values are generated, and we have generated a list of differentially expressed genes by applying an FDR cutoff of 10%. Okay. Now, what, right, what do you want to do after you have got the list, right. So, let us say you got 100 genes in the list, okay, and this is what we are going to talk about now, right. How do you actually interpret these results that you have got after this differential gene expression analysis? Okay. So, how do you interpret this data? Okay. So, we have this list of differentially expressed genes, and the questions that we are mostly interested in is that what do these genes do in the cell or the tissue, right. So, we want to know the functions of these genes. Why do you want to know these functions? Because they will give us insights into the biological processes. So, you can imagine this disease versus healthy situation, right. We are comparing this disease versus healthy samples, and we have done the differential gene expression analysis. We have identified a list of genes, and let us say we have identified 100 overexpressed genes in the disease sample compared to the healthy sample. Okay. And the question we probably want to understand is that what do these genes do, right, 100 genes, what are they doing? Are they in any way associated with the disease, right. So, whether they are causing the disease, right, or they are helping in disease progression, or whether they are actually doing certain functions, right, that kind of make the situation worse. Okay. So, the question that we want to answer often is that do these genes belong to specific functional classes, okay, and this is what we do by something called functional enrichment analysis. Sometimes these are also called gene set enrichment analysis, although there is a tool that is actually is called gene set enrichment analysis, or sometimes we call this as pathway enrichment analysis. Okay, these are kind of equivalent, but not exactly same. Okay, so let us now go into the functional enrichment analysis. Okay, and these are the questions that we ask here. Okay, are there specific functional classes that are over or under represented in the list of differentially expressed genes, right? So, we are probably asking this question because we want to understand the biology, okay, what is actually happening inside the cell in case of disease samples or in case of treatment versus control analysis, right. So, the genes that are overexpressed or the functional classes that are overexpressed or over represented, right, they are probably very important for the disease progression and so on. And the question, if we are analyzing disease versus healthy samples, right. So, this is probably one of the questions that will come to our mind, right. Do these pathways play any role in disease progression or response to a treatment or a drug or a therapy, right. So, maybe because these functional classes, they are kind of generating some sort of resistance to therapy, or if we are comparing, let us say, condition 1 versus condition 2, we are giving some stress to the cells. Are these pathways associated with stress response? They are helping in adaptation of the cells or the organism to that stress condition, right. So, this is again giving us the molecular insights, right. So, we understand how the disease probably is progressing, or we understand how the cells are responding to certain stress and how they are adapting to that kind of stress, right. So, we are getting through this analysis, we are now getting into the biology, right. So, we are getting more biological insight and we are trying to understand the molecular basis of disease or stress response and so on, right. So, depending on the question that you are asking, you are going to get the molecular insights. So, the first step in this process when you are going to do this functional enrichment analysis is to identify the gene sets, right. So, how do you define these gene sets, okay, and what do we mean by these gene sets? So, we want to, before we can do this functional enrichment analysis, right, we need to identify or associate each gene, right, to a function in the cell. Okay. So, you have list of 100 genes that are differentially expressed. For each of them, you need to have some association with certain functions inside the cell, right. So, this is the first requirement, we need to have this association, this information is required. Okay. And this one gene set, this consists of genes that are associated with a specific cellular function, right. So, let us say we have, let us say, glycolysis genes that are involved in glycolysis, you have ATP synthesis genes, right, you have TCA cycle genes and so on, right. So, you have these gene sets that are associated with specific cellular functions. Okay. And the other point that you also should keep in mind is that a single gene can carry out multiple functions, right. So, genes are pleiotropics, they often have multiple functions and thus they can be part of multiple gene sets, right. So, it is not necessary that all gene sets are unique, there can be overlap between gene sets, right. So, a gene that is involved in, let us say, salt stress response, responding to high salt concentration or high osmotic stress, right, it can also respond to oxidative stress, right. So, many genes have these kind of multiple functions and they can be present in different gene sets. Okay. So, one of the major tools for this gene set enrichment analysis, right, is the gene ontology. Okay. This is a resource where this kind of association information is present. Okay. Here is the link where you can actually find this information and these are the references. Okay. So, I will briefly mention what this gene ontology is, right, and of course, I will encourage you to explore more going through the resources that are present in the website. Okay. And you can also download ontology for any organism that you are working with, and there are many organism specific ontology that are available in the website. Okay. So, very briefly, gene ontology consists of a set of function classes along with relation between these classes, right. So, we have many functional classes and they are kind of related to each other, right. So, one is probably, let us say, glycolysis, that is something associated with carbon metabolism, right. So, these are functional classes that are related to each other. So, gene ontology will also preserve this information. Okay. So, it has this association of single genes to certain molecular functions, but also it has this information about relationship between functional classes. Okay. And this is a database of functional annotation of genes. So, you can you get this information, okay, which gene is involved in which pathway or which cellular process. Okay, so gene ontology is organized as a directed graph, as you can see, as you will see once you go into the website, okay, and each node represents a geo term or a functional annotation, and then edge it represents connection between geo terms, right. As I just mentioned one example, right. So, you can have these connections between different geo terms, okay. And it is also partially hierarchical. If you go and see the networks, right, this ontology, the directed graphs, you will see there is some sort of hierarchy little bit, right, where daughter nodes they describe more specialized function as compared to parent node. Right. So, again if you take example, let us say we have tryptophan biosynthesis, right. So, that will be a daughter node and maybe a parent node could be amino acid biosynthesis, right. So, you have these relationships or terms that are related to each other. Okay, so, in gene ontology, for each gene also you will find sub ontologies. Okay. So, what are these sub ontologies? So, there are 3 sub ontologies that are present. So, the first is molecular function, or in short MF, then you have biological process or BP, and cellular component or CC. Okay. So, you see these 3 terms in any ontology that you look at. Okay. And what do they mean? So, the molecular function, it means it actually specifies activity of a gene at the molecular level, right, what this gene actually does inside the cell. Okay. So, for example, this gene has kinase activity, okay, or maybe this gene is involved in the isoleucine biosynthesis. Okay. So, this is again the molecular function of the gene, actual molecular function inside the cell. Okay. So, this will be specified here. The second part is the biological process. Okay. So, here it actually will associate the gene with a larger biological process. Okay. So, for example, you can say the gene is involved in ATP synthesis. Okay. It does not specify the actual function, right, actual molecular function, which part of ATP synthesis process is it involved, that is not specified, but it is associated with ATP synthesis or maybe amino acid biosynthesis and so on, right. So, you see these are these actually associates a gene with a larger biological process, and again depending on your requirement, depending on your analysis, what you are looking at, whether you are interested in the actual molecular function or you are looking at the larger biological picture, right, depending on that you will choose the type of sub ontology. Okay. And the last one is the cellular component, right. So, this is actually associating each gene with its location in the cell or subcellular compartment, right. For example, a gene is located in cytoplasm or in cell membrane or mitochondria, this will be mentioned in this sub ontology. Okay. So, if you are looking at localization of genes, right, then you will probably look at the cellular component part. Okay. So, there are some geoterm elements that will be present in the ontology. So, the first one is the identifier, or sometimes we refer as geo ID, okay, and the name of the term. It will also mention sub ontology, right, MF, CC or BP, right, and it will have a description of the term, what this means, and it will also describe the relationships with other terms that are present in the ontology. So, for the annotation part, right, there is an annotation and this is organism specific. Again, you can look at the links, right, and this is regularly updated, and you see this is the most recent version that I have mentioned. You can go and see these annotations. These are organism specific annotation, because here the actual association happens, right. So, here each gene in an organism is connected to a specific function. Okay. So, these are the components of this annotation file you will find. So, you have like gene, RNA or protein names present, then you have association with a geo ID or geo term. It will have a brief description of function, very brief description, and then you have some evidence. Okay. So, you can now see, right, how you can combine ontology with this annotation, right. So, ontology gives us this geo ID, the term, and the explanation, the description of the term, right. Here here in the annotation you are associating the genes with the geo ID or geo term, and hence associating with specific functions. For the evidence part, right, you will see in the data, right, again there are a lot of details, right, how these evidences are collected, right, how they find which gene is doing which function in the cell. it could be experimental evidence, right, this has been experimentally determined in the organism, right, but sometimes this is not possible. So, it is also maybe inferred from expression pattern or inferred from computational analysis. So, all these things, right, are given in the annotation part, right. So, how strong the actual evidence is for that functional association. Okay. So, again you can go in the link and you can see the details. There is another database where you can find this kind of association of genes to biological processes or pathways. It is called KEGG, Kyoto Encyclopedia of Genes and Genomes. Again, here is the link and here is the reference paper. You can again look at the link and find out these associations. So, again you have association of genes with biological process and biological pathways here, and this is also available for a large number of organisms. Similarly, you have another database, is called MSigDB, molecular signatures database. Again the link and the references are given, and here you have organism specific gene sets, right, which again looking at molecular signatures. So, identifying which genes are involved in which specific processes, right. So, again you have something called hallmark gene sets. For example, you can have oncogenic signature or cell type specific signature data sets, right. So, some cell types will have expression of certain genes and so on, right. So, these kind of data sets are present in this database, right. So, you can again explore and see. And similarly, there is another database called Reactome. Again this is doing you have similar information, right, where you have association of genes with biological process and pathways. So, these are the references for this class, quite a few actually. So, again looking at all the databases here. So, in the first part of the class we have talked about FDR correction method, and compared to the earlier method, right, the FWER method, what we have seen is that FDR correction is more powerful, right. So, the power actually comes from this type 2 error. So, FWER commits a lot of type 2 error, whereas FDR reduces that type 2 error. So, what you see is that FDR can tolerate certain amount of type 1 error, but it also increases the type 2 error. So, where these two type 1 error, type 2 error are balanced now. So, FDR can detect the true positives more efficiently than the FWER. So, FDR allows for certain percentage of false positives in the results, as we have seen, right, and this is acceptable in many cases, including differential gene expression analysis. So, if you are identifying a large number of genes, if 5 or 10% of genes are false positives, they are not going to substantially bias our analysis or the downstream inferences, right. So, this is why we can tolerate this kind of false positives in many situations. Again, I have also talked about, right, where you will apply FDR method and where you will apply FWER method. Then we have moved on to the interpretation of DGE analysis results, and one of the first steps in this process is the functional enrichment analysis. And we have seen, right, we want to understand the functions of these genes that are differentially expressed, right. So, we want to associate these genes to certain molecular pathways, and we want to see whether certain pathways or certain processes are upregulated or downregulated, right. And this will actually help us gain more biological insights into the disease process, for example, or stress response or drug treatment, etc., right. And we have talked about some of the databases, right, that actually allow us to do this functional enrichment analysis. For example, we have talked about geo annotations, we have talked about KEGG, MSigDB, and we have seen, right, we have very briefly mentioned, right, how they actually associate these genes to their specific functions. Okay. So, and again there are different types of evidence that come into the picture. What is remaining is that we have not talked about the statistical test or the enrichment analysis itself. Okay. We have just talked about, right, where we can get these gene sets and these functional associations of genes, but what we want to do ultimately is we want to see whether any functional class is overrepresented, right, or underrepresented in the list of differentially expressed genes, right. And this is something that we will cover in the next class. We will actually look at the statistical techniques that will allow us to do this enrichment analysis and finally answer that question. Thank you.